This is Megan. Megan is a third-year biological sciences student at the University of Exeter. The reason we are concentrating on Megan today is because she is doing a research project as part of the marine biology department here in Exeter. Her project is part of a wider consortium to study the impact of multiple stressors on the marine environment. Stressors such as ocean acidification, human-caused noise, and many others. Megan's project studies the impact of changing temperatures and pollution on mussels, researching out in the field and back in the lab. Here, Megan is collecting mussels in Torquay, after already getting a number of other mussels at a different location in Devon. She is working with Dr. Kerry Lewis, a lecturer at the University of Exeter. Kerry specialises in marine invertebrates and is seen here imparting her ever-growing knowledge onto her eager student. Megan and Kerry need to collect about 70 mussels from each location for Megan's study. The number is this high as Megan is taking data from the mussels on location and then taking others back to the lab for further analysis. Megan is collecting the mussels in order to test the organism's reaction to changing conditions. There are two factors that Megan is studying, principally the changing temperatures that mussels are experiencing in their environment, however with an extra interesting twist on top: pollution. Our oceans are getting increasingly polluted, so researching what impact pollution could have on economically important organisms such as the mussel is vital. Torquay may not look as extreme as the Arctic or an African desert, but the conditions the humble mussel has to face every day are anything but easy. Without fail, in every 24-hour period, the mussel have to get through hours of low tide before returning to the relative calm of high tide. In order to survive, the mussel must keep its shell closed whenever it finds itself out of water, so as not to dry out. Megan is testing the pH of the insides of the mussel as she wants to find out whether there will be a significant difference between the pH at different temperatures, sizes, or pollution levels. When the mussels are out of water, they must keep their shell closed, which means they must breathe, or technically respire, using only the water that is contained within the shell. As there is no recycling of water, carbon dioxide will build up within the shell and within the mussel. As Megan is saying here, this causes a string of processes which results in the blood of the mussel becoming acidic. So Megan is testing to see whether the acidity of the mussel is increased or decreased in different conditions. The research will help us realise how future conditions will affect animals such as the humble mussel. There is a simple process to get the hemolymph from the mussels and then test it, as shown here. First, the mussel is opened and drained of seawater. The hemolymph is removed using a syringe, making sure to remove enough solution for testing. Megan will test the ambient pH of seawater to compare to the pH of the mussels. Once recorded, Kerry takes a sample in a capillary tube, which means the solution can also be tested later on in the lab if need be. After a few minutes, the testing and storing turns into a well-oiled and efficient machine. The mussels are safely transferred back to the lab in Exeter, where Megan studies the mussels once more. Here, Megan explains the process of the research in the lab.
I think I'm probably bringing back around 70 mussels for my lab work, and then so the mussels will be kept in quite a standard condition within the lab, and then I'm taking half of them from each location. So we went to Torquay the other day, so half of the Torquay mussels I'll take out of their standard conditions for 6 hours and put them in an extreme exposed condition. So I'm planning on doing 7 degrees exposed out of the water, and during the 6 hours, I'll probably sample every half an hour or 40 minutes, just like I was doing in the field, taking hemolymph samples, measuring the pH, storing them in the capillary tubes so I can then do TCO2 the next day. So once I've done the 6 hours exposed, I'm then putting the muscles back in the regular conditions, and for the next 2 hours, taking samples every 20 or 30 minutes, depending on how speedily I can actually do it. And then hopefully I'll see the speed or the rate at which they recover from being in the extreme temperature difference. So I'll do 7 degrees and I'll do a warm one, so I'll do 28 degrees as well. So hopefully I can get some good data looking at either end of the spectrum. And also I'm hoping to see that muscles taken from different locations, so Torquay and Star-cross, whether they recover differently. Because Torquay muscles, they're taken from the pier legs, they're not used to being exposed for as long periods as the Star-cross muscles are. So as I am forcing them to be exposed in these temperatures, I'm hoping to see some interesting stuff.
Megan's research and analysis is ongoing here at the University of Exeter. The results of her project, and projects of other students within the consortium, will help us understand crucial pieces of the puzzle of the impact of multiple stressors on the marine environment.